This conference will now be recorded.
Hello, Hello everyone. Am I audible?
Uh, yes, you are. Thank you.
Yes. Thank you. A very good morning, good afternoon, or good evening to one and all present here virtually with me today. Let me introduce myself. My name is Shirley Rose, Program Manager, Conference Secretary at Coalesce Research Group LLC, and I will be available for any of your queries or questions throughout the webinar to help you out technically and organize the webinar successfully. For the next 5 minutes, please allow me to give a brief description about our organization, Coalesce Research Group, and also the further instructions. Coalesce Research Group LLC is very delighted to welcome you all the speakers, supporting associations, students, and delegates present here virtually with us today. With the aim, mark the scientific excellence by facilitating conversations, sharing knowledge. Coalesce Research Group follows through the objective of setting standards in delivering excellence in the research among the researchers and industries, promulgating the research through conducting events and online webinars from various fields of science, especially from the stream of medicine, science, engineering, and technology. As a global scientific event organizer, we plan and carry out international conferences, meetings, seminars, symposium, and workshops, providing the best delegate experience, excellent networking opportunities, conversations leading to innovations, learning new opportunities. Just recently, we have also started our own platform for open access peer reviewed scholarly literature where all the content is freely available without any charge to the reader. As being a very new journal, we have coined up some interesting journals and have also formed the editorial board. And we will be very glad if any of our today's attendees, the speakers, students, or delegates could extend your support by clicking the journal and will be interested in publishing an inaugural issue at any of the journals according to your area of expertise. We will also send an email with all the details about the journal after the webinar. Do take your time to check the website. Thank you. Before we start with the sessions, I guess some of you will be new to the GoToMeetings app. Let me just explain you the important tools required for the presentation. On the GoToMeetings app or web page, you can find four icons at the bottom center: mic, camera, screen, and mic button. Click on that to mute and unmute. I request you all to stay muted while our speakers are presenting their research work so that it doesn't cause any disturbance to them. Camera button. You can use this button to share or unshare the web camera. If you have the option to share the web camera, you can do it and it is completely your choice. Screen button. You can use this button to share your screen. This is the main part during your presentation. After I call your name from my side, I will be making you the presenter and will give the permission to share your screen. Then only you will be able to share your screen. So once I make you the presenter, you can click on the screen button and you will find a pop-up window to share your screen. The last button is the end button. This is your fourth icon where you can use this to leave the session. On the top center, you can use the option to change your display settings. At the top right corner, you will have three icons: people, chat, and settings. In the chat option, you can chat with me or any other participants. For the settings, you can use this button to check. For the settings, you can use this button to check for the settings of your speakers and microphone. So as mentioned, kindly remain muted during the presentations so that the presenter don't get disturbed. And for any questions, you can ask the questions or you can send your questions in the message box. Well, let's not take much time. Finally, I would like to request you to please give us, excuse me. And now let's move into our sessions. The first presenter is the first presenter is Chikazi Kennedy Kalu from Jiangsu University, China. Hello, Dr. Chikazi Kennedy Kalu. Are you ready with your presentation?
Hello. I'm very ready. Good morning.
Good
Thank you.
morning. Thank you.
Yeah.
So I'll be making you as a presenter now.
Okay. Thank you.
You can share your screen.
Okay.
Yeah, I made you as a presenter. Kindly press the screen button to share your screen, please.
Okay. It's still on. It's still requesting permission. So I'm
Yeah,
not
give the
yet.
perm. So are you a MacBook user?
No.
No, okay.
HP.
Okay.
Yeah.
Please continue.
Yeah. Okay. So I see the button, but it's indicating that I need permission to be able to share. It said to share,
Okay,
ask
give
the organizer to make a presenter. Yeah.
yeah, I give the permission. Please kindly click on the share button.
Okay, I've clicked on it, but it's still barring me out that I cannot share. So it's still
Okay,
saying that I need the permission. Yeah.
1 minute. Let me check.
Okay. Thank you.
Okay, I made you as a
Okay.
presenter. Yeah,
Okay. It works now. Thank
yeah,
you.
yeah, yeah.
Okay. Can everyone see my screen? Just
No,
checking.
not yet, not yet. Yeah, I'm checking, but not yet. Your screen. Yeah. It's getting shared. 1 minute.
Okay.
Yeah, it's shared. Please make it a full screen.
Okay. Very good now.
Yeah, it's perfect. You can start your presentation now.
Okay. Thank you very much. And
Thank.
good morning, everyone. I'm Chikezie Kennedy Kalu, currently a PhD research student with the Jiangsu University in China. I'm seated currently in Nigeria, so I'm having a virtual study. My department is the Management Science and Engineering department, but my research focus area is technology and innovation management. Appreciate the opportunity to be here on this conference, and I'll be speaking briefly on the analytical optimization of X-ray mammography technique for increased benefits and safety using, of course, modern data analytics, electronics engineering, and artificial intelligence methodologies. Please, if you experience any delay in my slides, it could be the network. It's not so good at the moment, but I'll try to make the best of the situation. And if you have any challenges with my slides, please do let me know. Thank you. So as the title of my research indicates, the challenge or the problem uh, I was trying to analyze, research, and come up with solutions was usually for mammography and breast cancer checks and diagnostics, which is where mammography comes in very handy. There's been that challenge of striking the balance between safety of the patients in terms of radiation, harmful radiations, too intense X-ray radiations that could create problems both in the short term and the longer term for the patients, and also improving the quality of the X-ray images, because if we get the diagnostics very good, it also helps to profile better solutions or treatment for breast cancer. And it's been researched and noted by a lot of experts in this field. Early detection of breast cancer is one of the major ways to reduce it, as well as try to take it off, or try to resolve it with reference to the benefit for the patient. Secondly, we're looking at a situation whereby if this trade-off is achieved, how can we also ensure that the quality of our X-ray screening is improved? And this is where artificial intelligence comes in. I'll talk about that later, and then give more information with respect to this. The objectives, like I've hinted a bit, was to analyze, investigate common X-ray mammography techniques, terminologies, properties in terms of the system itself. And this is hovering around the field of medical electronics. Like the earlier speaker has mentioned, in line with this conference, this is looking at medical science, engineering, and technology, looking at how technology could be used to improve better diagnostics, analysis, and coming up with better solutions that could improve the medical field. And with respect to breast cancer diagnosis, for a safer, more effective, efficient procedure, using deep learning, artificial intelligence. Yes, deep learning is a field under one of the fields under artificial intelligence in terms of a methodology, electronic engineering, data analytics, and so on. Secondly, another objective or purpose of this was to be able to use data sets from approved stores, validated stores. This was a non-clinical research, but of course, data was taken from approved medical stores where clinical data, both historical and current, could be used to carry out this analysis. And this was where my focus on, and took up data to be able to analyze as well as come up with meaningful solutions and also observations. From X-ray mammography properties like the intensity, the energy of the X-ray, the K-edge energy, half value layer, and so on. These are items that were analyzed to ensure that a proper research is done and solutions are proposed. Can you see my current slide, please? Can I go on? Thank you. Okay. So right now we have the pictures, or before you, the experimental X-ray system setup, and also on the right you have the convolutional neural network system. The experimental setup, it's more like using breast equivalent tissue slabs to mimic a situation that could replicate what you have with human breast tissues, granular tissues, or fatty tissues. And for this experimental setup, the ratio of 70 to 30 was applied for a dense breast in terms of the ratio of glandular tissue to fatty tissue. And for a non-dense breast, the ratio of 50-50 was used, 50% glandular tissue, 50% fatty tissue. And from research has shown that glandular tissues have shown to be more susceptible to breast cancer. And so that is why going forward, as I will briefly explain, more focus in this research was channeled towards the denser breast, which is the one having 70% of glandular tissue and lesser, or 30% of fatty tissue. On the right, the convolutional neural network system, it's one of the applications or methodologies of artificial intelligence. Simply, what does it do? It's the ability to be able to create classified images with respect to data analysis, analyzing data images, and producing classifications intelligently. And this is quite key, because in modern X-ray mammography, we know about computer-aided designs, image designs, digital designs. The CNN comes in helpful because for modern data science and artificial intelligence system, it brings that extra touch of managing complexities, bringing up sharper quality images, as well as doing this with lesser resources. A bit of a limitation, which I've mentioned a bit in passing, is that this work was strictly based on computer simulation and algorithmic analysis. No direct patients in the hospital was dealt with, but of course, patients' registered data was used in carrying out this analysis. And of course, certain aspects of physiological or demographic factors which could affect patient's condition like age, location, climatic conditions, diet, and so on, were a bit kept aside because we wanted to, the idea for me was to focus on the aspects that affect breast cancer diagnosis in relation with the X-ray mammography system. So it's like bringing in engineering principles to be able to proffer solution to a challenging cancer situation that has troubled a large number of persons. The conceptual framework. This is the heart of the matter, whereby this is the conceptual design of the system on trying to optimize the mammography, X-ray mammography system. From what you can see the incidence radiation coming from the X-ray. You have automatic transducer sensors. Now that's a component, that's a black box. That's where we kind of have sensor systems, modern electronic engineering sensor systems that not only picks up electrical signals, converts radiation to electrical signals, and can also do vice versa. In addition to that, you have the AC to DC converters. AC to DC, what I mean is the analog converters to digital signals, and then it pushes it to the electronic impedance circuit. When it gets to this impedance circuit, this circuit is a branch of electronic system, circuitry system that is able to work on this electrical signal and then come up with signal equivalents that are needed in reference to the analysis. The reverse, in reverse, it converts it back to analog signals, which now are fed to the metal filter for the X-ray mammography system and then to the tissue. The idea behind all this is, as experts have researched on this, and I have also researched on this, there is a key problem with the intensity of radiation. The filters, if we remember, are very much important in reducing the intensity of the X-ray radiation, but they do not reduce the energy of the system. And this makes it interesting. We are worried about the intensity of the X-ray, and rather than leave it all to the metal filters, which sometimes can be cumbersome in getting the exact thickness, the exact layers, and also with time, the metal filters experience wear and tear. Electronic instrumentation can come in to be able to beat down this intensity or increase the energy if we want to, because X-ray mammographies are generally low energy systems. So this electronic system is coming in to help out to all do what? To optimize the metal filter processes of beating down the harmful radiation, intensity of radiation that comes to the human tissue. Just below you on the left, you see the circuitry. That's the circuitry description of the X-ray impedance circuit. That's the branch arrangement. And then on the right below, you have the typical CNN architecture for mammography, as I've briefly explained. You see the input image. It classifies it whether it's normal breast cancer we are looking at, or benign or malignant. Next slide. For the method, I tried to summarize it here to a slide or two, whereby the equations that were used to come to this, to carry out the research and also come to meaningful conclusion. On the first left, you have the transmission power, which is the equivalent radiation power from the X-ray. And from the principle of energy conversion, that power too is the same power coming into the circuit related to the voltage of the X-ray and also the total impedance of the circuit as described in the previous slide. P2 is the power output now from the metal filter that goes to the human tissue after it has been resolved or after it has been analyzed by the circuit, and the impedance in that case is Z2, which is the output impedance of the circuit. You have II, which is the incidence current, and you have IZ, which is the current from the metal filter. You have the energy, and we know that the energy is related to the incidence by the speed of light, which is C, and there's also a relationship. So all these calculations were done to be able to come to a point whereby incidence radiation from the X-ray is converted to electrical signals, which the sensor or the electronic system could work on. And after the electronic system performs its operation, this is now reconverted and fed to the metal filter and then to the tissue. In this way, intelligently and electronically, we're able to handle the incidence radiation. Not just only handle the incidence radiation, we're also able to provide a flexibility in terms of being able to tune whether the incidence radiation or the energy of the signal. And I believe that for researchers, for radiologists, for the medical science, this will be helpful because it means we can vary the incidences and not only be fixed on a metal filter that is related to the incidence radiation we are trying to reduce. These are also more equations, the maximum photon energy relating to the energy from the filter and also the energy from the field radiation. Like I've earlier explained, the last equations, the last two by the right-hand side has to do with the pooling layer of the CNN architecture system. Pooling layers are necessary because they help to minimize the size of the image, and this is necessary because we are talking about loads of data, complex images. So this pooling area helps to minimize as well as retain the quality of the images. In this way, a better quality image comes up, and at the same time, it is well managed. And importantly, in line with the modern green solutions that have been emphasized, it reduces the energy consumed to produce such excellent images. Some results were gotten, important results, and I have a few shared here. The first one on the left has to do with the plot of a dense breast imaging technique and energy relationships. Now, what was done here was the different mammography technologies, whether it's the modern contrast enhanced system, or the tomosynthesis system, the 2D full digital mammography. All these were analyzed in combination with specific metal filters, and some results came up on this, which we can see. We noticed there is a relationship, of course, noticeably with the filters and also how much energy is absorbed. Noticeably too, we see that the combination of the filters and specific technology also varies, meaning that for the modern contrast enhanced system, a filter with a lesser K edge energy could be used optimally and efficiently, yeah, rather than using a filter with a very large cutoff energy and thereby limiting the efficiency of the system. On the right, we have the plot of the dense breast. Specific attention was paid to the contrast enhanced system, which is a modern technology which combines digital mammography as well as an enhancement mechanism which is able to improve the quality of the image. And we can see that the intensity with the circuit impedance does drop, which is expected and which we need. But at the same time, we can also see that the impedance of the circuit tend to increase with the intensity. What that means is, depending on the intensity that we need, the target intensity, we can always vary our circuit impedances and obtain the results that we need. Also, additional results are shown here in terms of the dense breast, the CSM technique, energy and circuit impedance relationships, as well as the half value layer. The half value layer is of a breast or a material is the layer at which the intensity of the radiation is reduced by half. Now, this is also important because if we are creating an integrated system such as this, we do not need to use metal filters that could be so consuming in terms of energy, but we can also integrate with the electronic. Knowing the half value layer, the electronic system can beat it down and also ensure that the metal filter we use is not one that is challenging to obtain, as well as making sure that our target or our X-ray mammography is very much accurate and flexible to be able to vary the intensity. For the artificial intelligence system or the CNN methodology, what it does, like I've explained earlier, is to analyze images, creating better images in terms of quality, using up less energy, as well as doing it in a much more efficient way. So a research was done on this, comparing pooling layers. I've explained what the pooling layer does. The first one on top uses one pooling layer, and the metric of conversion or the metric of comparison that was used here was the validation accuracy. Now, the validation accuracy is an important metric when it comes to measuring how accurate or how quality our classification, or in this case, our image comes out. Now, how is that done? Usually for artificial intelligence system or for intelligent learning system, our data is split into training sets and also test sets. The training set is used to train the system to be able to understand the type of data, and the test data is now then used to validate how accurate our data is. And this is where the validation accuracy comes in place. The validation accuracy noticeably increases with the number of pooling layers. And of course, for better pooling layers, for better validation accuracy, there should also likely be an increase in data sets. This helps us understand that for such an integrated system to be able to come up with better accurate images for better diagnosis with regards to breast cancer and X-ray mammography, such a system ensures efficiency by making sure we use as less resources as possible to obtain big and very important results. Discussion and suggestions. Well, a few items were pointed out from this research work. Of course, the contrast enhanced spectral mammography, the CESM technique, confirms to be the most efficient, especially in a dense breast condition, which was our focus. Other techniques or systems, this research shows that they could be improved in terms of efficiency when they are combined with the right set of filter. Like one of the results I showed earlier, we saw that research showed that with different applications or combination of filters, we see improvements in results, even with older systems that may not have given as much quality in terms of image classification. Furthermore, the electronic system shows good promise in terms of, like I've mentioned earlier, this gives us flexibility. We are not just stuck with a target intensity or energy using the metal filters alone, but now we can vary it with intelligent electronic circuits that can produce different levels of intensity as needed for a specific purpose. This increases safety, this increases better benefits, and this makes it something that could be improved on for more excellent results in the future. Additionally, in terms of the artificial intelligence system or the convolutional neural networks, the number of pooling layers needed to achieve the quality metrics was also investigated and proposed. Like I showed earlier, equations were developed that confirmed this mathematically, that pooling layers have a kind of a direct relationship with how quality or the validation accuracy of our system. These are the suggestions and recommendations. For the CNN system, perhaps to improve further on this, even deeper layers of processing can be experimented. The CNN can always give more performance because the more layers, the more performance. But at the same time, we're looking at a system that can be efficient. So there could be limits to how much layers we want, but depending on what we want to achieve and how much data we need to analyze, then deepening the layers of processing also improves the quality of images. Furthermore, in terms of the filter combination and the electronic designs, much more can also be added to this in terms of filter combination, electronic circuitry combination. The electronic circuitry designs also works hand in hand with the K-edge energy cutoff of the metal filter. Uh, think of the K-edge energy cutoff as perhaps, if I digress a bit, when we talk of electronic communication, there's something we call electronic filters that filter signals, low pass, high pass, and band pass. So what that indicates is certain filters cut off any energy beyond a certain level. Certain filters do not allow energy beyond a certain level. Certain filters, the band pass, very flexible, allows energy to flow within a range of energy settings. Uh, that's this my last slide. That's all from me. And I will just end with this bit of quote from Edward Teller, which says that the science of today is the technology of tomorrow. And from my end, permit me to add a little bit to that. And the bridge between both, of course, is humanity. Thank you for your audience. Please, you are free to ask questions, or if you want to add one or two items to this, you are free to do so within the time I'm allotted. Thank you.
Thank you, Dr. Shaisi Kennedy Kallo. That's a wonderful
Yeah,
presentation. Uh,
yeah.
does
Thank
anyone
you.
have any query? Yeah, there is one.
Okay.
Hi, and I
Hello.
have one question. Hello, and I think there are many
Oh, yeah.
deep
Hello, Shan.
learning. Hello, there are many deep learning methods or models, and why you choose the CNN?
Hello, Shan.
Can you hear
Okay.
me?
Thank you. Yeah, I can hear
Okay.
you. Is that all your question? You said there are many deep learning models. Why did I choose CNN?
Yes.
Okay, thank you very much. I think in this case, the reason for CNN comes. Okay. The reason in this case, firstly, CNN has to do with image classifications, and in dealing with this X-ray imagery, it was of one of the best in terms of the best when it comes to handling images and providing intelligent classifications, especially when it has to do with breast cancer images that could be normal, benign, or malignant. Secondly, the CNN process provides a bit of flexibility in in the sense that it could with variation of data sets and also variation of parameters, it could be able to carry out this analysis and provide accurate results. Thirdly, it's quite fast in terms of how it can carry out these results and how efficiently such classifications could be done. So, on the top of my head, that's the three main key reasons, in addition to possibly others, why this was chosen. Did I answer your question?
Okay. Thank you.
You're welcome, Shan. Thank you.
Okay. Okay, thank you. That was a wonderful presentation by Dr. Shaigezi Kennedy Kallo. Does anyone have any
Yeah.
questions? Further questions? Okay, there are no questions. Let's move on to the next presentation. Thank you, Dr. um Kennedy Kallo.
Thank you so much for the organization and thank you for inviting me to this conference. I wish you all the
Thank,
best and I'll be happy to work more with you in the future. Thank you. Bye.
thank you, thank you. Glad to hear that from you. Thank you so much.
Yeah. Bye. Thank you.
Okay, let's move further. Let's move on to the next presentation by Shan Liu, from the Hong Kong University, Hong Kong. Hello, Dr. Shan. Are you ready with your presentation?
Yes, I'm ready.
Okay, I'll be making you as a presenter. Please click on the share button, please.
No, it's still green.
Yeah, I'm making you as a presenter.
Okay.
Yeah, I made you as a presenter. Please share your screen.
Yes.
Yeah, your presentation is visible. Kindly make it a full screen.
Okay.
Yeah, it's perfect. You can continue with your presentation.
Okay, thank you everyone for listening to my presentation. Here I will share with you the title. DPP2 is a potential cancer stem cell marker in hepatocellular carcinoma. Firstly, there is some background information. The liver cancer stem cells are only a small subset of the HCC cells that has some embryonic stem cell or the liver progenitor cell features. And those cells always contribute to the different treatment, the resistance, and the tumor recurrence. There are some famous cell surface markers of these cancer stem cells, such as CD133, epicam, CD19, and CD44. They are um, they are discovered by the fluorescence activate cell sorting method, and recently the single cell RNA sequencing also provides some mRNA level of these stem cell markers. Such as for the epicam and LGR5. These are some stem cell related signaling pathways such as TGF-beta pathways, Wnt beta-catenin pathways, and status 3 pathways. And this part is our previously colleagues' work. They established an in vitro hepatocyte differentiation model using the different different culture median and growth factors from the embryonic stem cells to the endoderm cells and to the liver progenitor cells and then to the premature hepatocytes. And these two are two samples of HCC patient with the adjacent normal tissue and the HCC tissue. And from this panel, you can see this stemness-related genes and epithelial cell-related genes has a distinct expression pattern. Um, for example, the OCT4 and SOX2 has reached the peak expression at the ESC stage, and the ALB and CYP3A4 reach their peak only in the normal tissue. So, as the name indicated, developmental pluripotency associated protein 2, the DPPA2, also called, I will call it DA2, it will has a similar expression pattern as ESC stage cells. And so you can see it has reached its peak at the embryonic stem cell stage and then dramatically downregulated through this panel of different development stage. And you can see in HCC it also has very low expression. I think it's due to there is only a very small subset of the DA2 positive cells, and so the average expression of the mRNA level is very low. And this cloud work a word cloud shows some co-occurred genes in the published paper. They are SOX2 or 34 nalog. They all validate DPP2 is stem cell stage related genes. And this gene is mainly expressed in the nucleus. This part is our own patient data because the TCGA data and the ICGC data, there are only a very few patients, less than 10, has this DA2 expression, so we did not use this data. So in our own patient data, we can see that although the DA2 has very low expression and very low average expression, it has significant difference between the HCC and the paired normal tumors. This IHC result also confirmed that only a very small subset of HCC cells can have the DA2 nuclear positive. And this is clinical pathology features and the prognosis of OS. We can see upregulated of DA2 can predict poor OS. And based on this OS, we define the cutoff for the DA2 normal or upregulation. So we can see the upregulated DA2 is related to a more advanced stage of a more advanced stage and multiple tumor number and poor differentiation stage, and it is also related to the presence of tumor thrombus. And in the CCLE database, we can see that most types of the HCC cell lines have no DA2 expression except for the HU7. And this is our own qPCR data also confirm this. The MIHA is the lung tumor immortalized liver cells. It has almost zero expression of DA2. The FACS flow cytometry result also support our findings. This is the immunofluorescence result, also support our findings. And then we want to see if DA2 can have a correlation with CD133, which is a popular cell marker of the liver cancer stem cells. And we can see that the mRNA level has a positive correlation between DA2 and the CD133 in the CCLE database. Also, the RNA level is significant, enriched in the CD133 positive HU7 cells of the DA2. And then another data set also shows that the CD133 positive cells can have higher mRNA expression. And then we want to see if DA2 can perform some regulation to the functions of cancer cells. First is to the overexpression of this gene in the MIHA and HEP3B cells, just as our previously described. And we can see the confirmation of the MIHA and the HEP3B overexpression in both Western blot level and the immunofluorescence level. And this is the overexpression of function of DA2. We can see the XTDSs did not infected by the DA2. So, but the sphere formation and the soft agar acids show that it can enhance the stemness of HCC cells without affecting the proliferation rate. The ALDH activity axis is higher in the stemness cells than the cancer cells. So we can see this is a baseline. We can see upon overexpression of DA2 can upregulate the LDH activity in MIHA cell and HEP3B cell. This Western results show that the embryonic stem cell marker SOX2 analog has upregulated by the DH2 overexpression. This is an in vivo assay that called the limiting dilution with different lower and lower tumor cell injection, and we can see the tumor proliferation rate and the size is significant higher in the DH2 group, overexpression group. And then the migration and invasion assays also confirm that DH2 can enhance the migration and invasion. And the Western result shows the EMT markers also have the expected trend. This is a 12 injection of DH2 into the mouse, and after 3 months, we can see their metastatic lung nodules found in lung lobe. And for the drug resistance, we did the flow cytometry. We can see based on the baseline, the DDP and 5-FU and sorafenib resistance is occurred when we overexpress of the DH2. This part is for the lockdown functions. The HU7 was choose to knock down, and we can see the sphere formation, and it was impaired related to the stemness, and this time the XTD assays were impaired by the knockdown, not the same as overexpression. And also the colon fossa formation, and also see the impaired function of knockdown of DH2. This are migration and invasion result. Also have the similar trend. Then for the drug resistance, for the previous 3 drugs, we can see the XTD assays shows the similar trend, but only sorafenib and the DDP was verified by the flow cytometry. And this is in vivo assays. First is the subcutaneous injection of HU7 cell into the mice. We can see the significant difference. And this is a 12 injection of HU7 cells. Also, it is kind of significant. And then the picture C is also tropic tumor cell liver injection. This is still very significant. And when we do surgery and got out of the liver, we see the same result. And this is in vivo limiting dilution result also show the tumor formation capability was impaired by the knockdown of DH2. So it seems that DH2 has a relatively strong functions, strong capability to induce some stemness-related function. But how is the mechanism between the regulation of DH2? First, we do the RNA-seq of HEP3B cells. Under these 2 conditions, we got those 7 stemness-related genes. They were very top and among the top 20 upregulated genes. Among them, KRT7 is a liver progenitor marker, and it is an interesting indicator. And this is enrichment analysis. The KG and the reactant analysis of HEP3B shows that they enrich in the G protein couple receptors pathways. You can see here, and here. And also PI3K-AKT and CAMP signal pathways is downstream of these GPCR's pathways. The similar results also seen in the MIHA enrichment analysis. The GO enrichment analysis also shows the similar result. So this RNA-seq data show us that the stiffness-related genes were very significantly upregulated in DA2 overexpression, and some non-top upregulated genes can be enriched in GPCR pathways. And we can look at this upregulated stiffness genes. They are involved in this some special pathways. We also did Wnt pathways. We in experiment show it can be influenced by the DA2 overexpression. Data not shown here. So it seems that many pathways are activated. So how can DA2 regulate this? There's so many pathways. Then I did the protein-protein interaction, and you can see these three proteins. They are all mainly located as the nucleus, and they were related to the chromatin modify and the histone modification. So my next step want to see if DA2 can change the overall state of the chromatin and the histone. If the histone is changed. So this is my last figures. Thank you.
Thank you, Dr. Sean. That was a wonderful presentation. Does anyone have any query? Okay, there are no questions. Let's move further. And once again, thank you, Dr. Sean, for your wonderful presentation.
Thank you.
Okay. The next presentation is by Dr. Manfred Diop, from Holistic Center, Switzerland. Hello, Dr. Manfred. Are you ready with your presentation?
Hello?
Hello, Dr. Hello.
Yes.
Hello. Hello,
Hello.
Dr. Hello.
Yes.
Are you ready with your presentation?
Yes. Can you upload my PowerPoint?
Sure. I will check that with my program manager and get back in a minute.
Yes.
Yeah.
Yes. Hello. That's me.
Hello, Dr. Manfred.
Yes. Thank
Yeah,
you
this is like,
for uploading.
yeah, thank you. Please continue with your presentation.
Yes.
Yeah, please say the next slide so that we will be moving the next slides. When you say
Okay.
next, we will be moving the next slides.
Yes.
Thank you.
Next slide, please.
Yeah.
Okay. A main problem in routine cancer treatment is regularly that the untargeted chemotherapy kills the sensitive cells but does not reach the resistant cells clones. These can then multiply at will, and metastasis can and will develop. The literature on cancer therapies is full of regimens that produce improvements in survival rates of a few months only. However, rarely someone writes about the possibility of achieving a cure. This can be implemented in reality by means of the individualized therapy. One attention for cancer therapists: poking the tumor on the occasion of a tumor biopsy may also promote metastasis. There we have to be careful. Next slide. Unfortunately, it is a reality that although oncologists repeatedly promise personalization or individualization instead of regimens, this hardly ever happens in routine. And it is only these conditions, however, that can provide a leap forward in cancer therapy. Tumor-specific and individualized chemotherapy combined with other therapeutic approaches that promise to be successful. Next slide. For this purpose, a laboratory method exists. It is an in vitro method, the liquid biopsy. We have been performing it on every cancer patient for more than 2 years. To do this, 20 milliliters of blood is sent to a specialized laboratory in Greece. The homepage is shown here. There, the cancer cells are identified, selected, and multiplied. With their help, one can now recognize the type of cancer. One can test all existing therapeutic agents for their effectiveness on the cancer cells, and also test about 70 potentially cancer cytotoxically effective natural remedies. It is shown here the homepage of Wikipedia concerning liquid biopsy. It is a worldwide accepted method. Next slide. The result lists are rankings of effectiveness in killing cancer cells. Individual therapy can now be put together from the best performing chemical and natural agents. Of the chemical agents, for example, the ancient cisplatin has proven effective very often. Of the natural agents, angiostop made from sea cucumber is very effective. But Artemisia annua, the annual mugwort, also proves its abilities, as does high-dose intravenous vitamin C. We put intravenously 30 to 60 grams of vitamin C. We have made the experience that individualization allows a reduction of the dosage in chemotherapy to about 50%, the so-called low-dose chemo. Next slide, please. As a third pillar of our therapy, We use intracellular enzymes from the company Cytoceatec from Italy. The homepage is shown here. The enzymes are used by means of tumor-specific intake protocols. The enzymes do not have a cytotoxic effect, but convert the lactate metabolism, which is typical of cancer, and back into oxygen metabolism, as the enzymes are working intracellularly and even intracell-nuclearly. So, the cancer cells can thus be re-socialized. Enzymes have proven their abilities in cancer studies at the Tor Vergata University in Rome within a study of non-small cell lung cancer patients. The patients receiving only chemotherapy all died. The patients receiving enzymes parallelly survived. The literature is shown here. Next slide. Our cancer therapy concept is thus as follows. Liquid biopsy, the laboratory RGCC in Greece, with identification of individually effective chemotherapeutic agents. The three best ones, most effective ones, are used, if possible, intravenously as low-dose chemo. Second, application of the 6 most effective natural remedies. Third, parallel implementation of enzyme therapy according to the protocol of the cancer type, via as many access routes as possible, as the enzymes can be used orally or intravenously or into the skin or wherever, even close to the tumor. This cancer therapy has improved our success rate by far, and it is a logical therapy, not an ideological therapy, and not a radical one, but a rather soft one. The hairs of the patients are not lost, and the patients can go over the therapy without many problems. So, next slide. Thank you for your attention. I'm looking for questions if there are any ones.
That's a wonderful presentation by Dr. Manfred Diop. Does anyone have any query? Hope there are no queries. Let's move further with the presentations. Thank you, Dr. Once again, thank you, Dr. Manfred Diop, for your wonderful presentation.
Yes, thank you too. Bye-bye.
Bye. Okay, let's move further with the presentations. The next presentation is by Mahjor Saumicha from University of Granada, Spain. Hello, hello, Dr. Mahjor Saumicha. Are you ready with your presentation? Hello, Doctor. Hello.
Hello, am I audible?
Yeah, you're audible. Are you ready with your presentation?
No, no, no. I am Subharanjana. Actually, I have one question
Yeah, please.
to sir. Actually, this is nice presentation. Actually, my audio some technical issue.
Okay,
I unable
you
to
wanna
connect.
ask question to Dr. Manfred Diop?
Yes, 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 yes.
Okay, okay.
Uh,
Hello, Dr. Manfred Diop.
Okay.
Hello, Dr. Manfred Diop. Yeah,
Yes,
Dr.
I'm here.
Subarajan Jena want to ask you a question regarding your
Yes.
presentation. Yeah,
Okay.
Dr. Subarajan Jena, please continue with your question.
It's a very nice presentation, sir. Sir, I have one question. Actually, sir, I am working in cancer registry in India. Sir, this liquid chemotherapy, liquid biopsy, it is very new test, or it is already happened in across the globe. In India, also liquid biopsy is available, or it only Switzerland?
Yes, I did not understand very well, but as I understood, I can agree with you. We are in the same region of medicine.
Okay, sir. Okay, sir. Okay. No,
Yes.
sir. I
Thank
have
you.
actually my question is this liquid biopsy. This liquid biopsy is currently available in all over the globe, across the globe, or only it's available in only high income countries. My question is this, that sir, liquid biopsy test it can it is undertaken in India or other low and middle income countries, or only it is conducted in only high income country like Switzerland.
Yes, the methods I gave as a presentation are available all over the world. The Greek laboratory using liquid biopsy accepts blood samples from all over the world, and the enzymes which we use from Italy can also be purchased all over the world, and you only have to contact the laboratory and the factory in Italy of Mr. Ferrorelli, and you will get your local contact email. Okay.
Okay, sir. Thank you, sir. Thank you so much.
Yes, thank you.
Hello, Dr. Saumicha. Can you hear me?
Yeah, good afternoon.
Good afternoon. So, are you ready with your presentation as the next presentation is by you?
Yes, now or
Yeah.
after?
No, no, now only. I'll make you as the presenter. So, please click on the share button to share your screen, please.
Okay.
Okay. Hello. Yeah, did you get the. Hello, Dr. Saumicha.
Hello. Do you hear me?
Yeah, did you. Yeah, yeah, I can hear you. I made you as a presenter. Can you please share your screen?
Please, 1 minute. I'm sharing the screen.
Yeah. Yeah, please continue. Okay. Make it a full screen, please. Yeah, your screen
Yes.
is visible. Make it a full screen.
So I can begin?
Yeah, no, you make it a full screen. Your screen is visible, but make it a full screen.
Okay.
Yeah, it's perfect. You can continue with your presentation.
Okay. Hello everyone, my name is Sumisha Mahjour. I am a scientist from Algeria and a member of the Natural Product Chemistry Research Team at the Organic Chemistry Department, University of Granada, Spain. As a researcher in the field of natural product chemistry, my work focuses on the exploration and investigation of bioactive compounds derived from natural sources. By harnessing the power of nature, we aim to discover potential therapeutic agents for various human diseases, particularly in the field of oncology. It is an honor for me to address you today at the Third European Congress of Cancer Stem Cell and Oncology Research. I'm excited to share with you the remarkable findings of my research titled First Synthesize and Anti-Cancer Activity Evolution of Novel Podocarpine and Abiantine Derivative. The second slide. Cancer remains one of the greatest challenges of our time, and extensive research efforts continue to be dedicated to combating this devastating disease. My team
Hello,
and I,
hello, doctor. Hello, Dr.
yes,
Saumicho. Sorry for the interruption. Yeah,
yes,
there is a poor network connection from your side, and there is a lot of disturbance in your audio. Can you please check your microphones and your network connection, please?
okay.
Yeah.
Hello, do you hear me?
Yeah, I can hear you. Yeah,
Yes,
make it a full screen, please.
yes. And now, we understand the urgent need for effective treatment against cancer, and our focus lies in developing novel methods to synthesize molecules with potential anti-cancer activity by harnessing the power of chemistry and pharmaceutical sciences. We aim to contribute to the discovery and development of the new therapeutic options. In our investigation, we have chosen to concentrate on the remarkable potential of podocarpine and abetane derivatives in the realm of anti-cancer research. These natural compounds belonging to the deuterapoid family have been isolated from various terrestrial plant sources. They exhibit a diverse array for integrating biological activities, capturing the attention and interest of the medicinal and pharmacological communities. It is with a great delight that I stand before you today to present the findings of our research, divided into two key parts. In the first part, we focused on the pioneering synthesis and biological evolution of novel podocarpine derivatives. Among them, we successfully synthesized and characterized 12.19 dihydroxy, 13 acetyl, 8.11.13 pyrrolidine, a compound previously unexplored and isolated from Gaultheria yenesis. Additionally, we achieved the synthesis of abetane type 2 terpenoids derived from the natural lipidine deuterapen transcommunic acid. The second part of our study delves into the evolution of an anti-pyrrolide activities of these newly synthesized compounds, along with previously reported synthetic precursors. We conducted extensive experiments using well-established human tumor cell lines, namely NCF7T84 and A549 by closely examining the impact of these compounds on cell proliferation. We aim to gain valuable insight into their potential as potent anti-cancer agents. In the course of our research program, aim to prepare new bioactive compounds from transaminic acid 7 isolated from Coalesce sempervine 2. We are interested in developing a new strategy for synthesizing C18 functionalized abetane phenols, such as A5 isolated from Cretomeria japonica 3.4 and the bioactive 7.6.7 dehydroreductive 6 19 hydroxy 4.5. The aromatic ring of target compound is formed a new series of podocarpine and toluterin derivatives, including the natural podocarpine 23, has been synthesized and evaluated for in vitro antiproliferative potential against different human cancer cell lines, NCF7, T84, and A549, and the antiproliferative cancer effect was induced by compounds 23, 27, and 28 with IC15 10 millimite and into 27 or 3 cell lines 23 and 28. No correlation with log P values was observed. The totalen 27 showed limited toxicity in normal PBMC and appeared and exert an antiproliferative effect in A549 cells through a mechanism that involves the induction of apoptosis mediated by an increased Bax BCL2 ratio. The result of the present study indicated that compound 28 might be useful as antimural agent. Further studies are required to locate and cellular molecule element involved in its effect, and the levels of activity, toxicity, and pellicle models. Thank you for listening. And thank you also for your time.
Thank you, Dr. Mahdav Saumicha. That's a wonderful presentation. And does anyone have any query? Hope there are no queries. Let's move forward with the further presentation. Hello, Dr. Saumicha.
Yeah, hello.
Yeah, please unshare your screen, please.
Okay.
Yeah, thank you. Okay, let's move further. Yeah, let's move further with the presentations. The next presentation is by Dr. Subarajan Jena from Institute of Medical and Some Hospital, India, Hello, Dr. Subarajan Jana.
Yes, yes, madam. Good afternoon.
Good afternoon. Good afternoon, doctor. So, are you ready with your presentation?
Yes, yes, I'm ready.
Yeah, I'll make you as the presenter. Kindly click on the screen button, share button, please.
Yes, yes.
Wait a minute. Yeah, I made you as the presenter. So, kindly click on the share button and share your screen. Yeah, your
My
screen is.
slide is visible.
Yeah, your PPT is visible. Yeah, it's perfect. You can continue with your presentation.
Okay. My slide title is Integration of National Cancer Registry Program with Ayushman Bharat Digital Mission in India, a necessity or an option. And this is my presentation outline, which include introduction, objective, methodology, result, and conclusion. And as you know, cancer is an emerging major public health problem across the globe. And according to the World Cancer Report 2020, the cancer burden is increasing unequally, impacting among low middle income countries. And according to that report, cancer is either the first or second leading cause of premature death among people aged between 30 to 69. And India rank is in second rank. And that report estimated 7.8 lakhs cancer death and 1.16 million new cancer cases have been reported from India, which are population 1.35 billion. In India, in lifetime, 1 in 15 India have risk of death in cancer, while in among 10, 1 person, 1 Indian will have developed new cancer. So, uh, as we know how cancer is day by day increasing over the globe. So, the cancer registry is one of the essential components and plays a critical role in cancer control program. Without proper data, without proper patient data, we cannot control the cancer control program in any country. The National Cancer Registry Program, basically in India, which has begun in December 1981 under the Indian Council of Medical Research. And now, currently in India, we have 236 hospital-based cancer registry and 36 population-based cancer registry. On September 27, 2021, the Honorable Prime Minister of India inaugurated the Ayushman Bharat Digital Mission. Means all the data of patient will be captured digitally in India. It's a very beautiful step by Government of India. And initially, it is being piloted in 6 union territory of India. Now it has implemented across the country. And our objective is in articulating Indian National Cancer Registry Program, we have attempt to assess the current mission, which is National Ayushman Bharat Digital Mission, with that critical objective. And the methodology, we review the literature from various databases like PubMed, ProQuest, Web of Science, Embase, and by using various keywords like cancer registry, management, digital mission, Ayushman Bharat, we use that keyword to source the available research paper. And what we found, basically, if we'll say about what is the landscape of Ayushman Bharat Digital Mission in India. Uh, Under this mission, every citizen will receive 16-digit unique health ID, uh, which will be linked with Aadhaar card. Aadhaar card, it is a identity proof. You can say identity proof of our country, India. So it will be linked with Aadhaar card, and every person, every citizen of India, they will get 16-digit unique health ID. And that after integrating that health ID in the Ayushman Bharat Digital Mission, when the person will open one ID, then it contain all the health record, including medical tests, diseases, and previous prescription. All the record will kept in that digitally, and all the information will kept confidential. It will not share with anybody. The primary purpose is, of this mission to reduce the out-of-pocket expenditure and ensure the realization of universal health coverage. The mission goal is to create digital health system that will boost India's healthcare system efficiently, effectiveness, and transparency. So, to reduce the economic burden of six group of common and high-cost disease, including cancer, the government of India initiated the Ayushman Bharat scheme 2018. Now they have initiated Ayushman Bharat Digital Mission, but in 2018 they have initiated another mission which is Ayushman Bharat scheme. In Ayushman Bharat scheme, all the low middle income family they will get up to 5 lakhs of insurance scheme. So, however, that health in India, basically, what is the major problem? Our policy, basically, healthcare policies, it is completely a state subject. Means without state permission, the central government of India they cannot do anything. So, as health is a state subject in India, so Ayushman Bharat has not yet been implemented in few states like Telangana, Delhi, West Bengal, and Odisha. So, as you know, Ayushman Bharat is still not implemented. Now now government of India is implementing Ayushman Bharat digital mission. So here we found what is the importance of digitalization in cancer registry. Actually, digitalization play a vital role in proper functioning of cancer registry. Uh, uh, how it will improve the treatment information, data entry process, patient that accessibility of patient data, completeness of cancer registry data, to follow up the tracking patient data, data retrieval. So like that, digitization play a very vital role in cancer registry. A study which was conducted in China, which found that modern digitalization technology help cancer registry to manage patient and clinical data. How? Because in China they use mobile and digitalization in their hospital, and by the help of their digitalization, a healthcare provider uh, easily track the patient record, clinical progress, treatment history, and various tools they can use easily. So, meanwhile, it is also the digitalization use of digitalization cancer registry. Basically, I am saying about low middle income country and basically India. It will help to both researcher and clinical practice because they can easily easily all the data within one very few times. So why I'm saying about integration of National Cancer Registry Program with Ayushman Bharat Digital Mission? Why it should be integrated? What is the necessity? So National Cancer Registry Program report, which was published 2020, it recommend linkage of cancer registry with Ayushman Bharat or other insurance scheme, mortality database, and healthcare management information system at the national and local level to improve cancer statistics. As you know, cancer registry it is the gathering of patient data. So without linkage with other various insurance scheme and more death mortality database, we cannot get accurate data of cancer patient. So without accurate data, we cannot implement also cancer control program in any country. So, though the cancer registry has started in India basically cancer registry has started in 1981, but still it has not completely established across the country. Still, many states it has not established. So, further, as per the report, the death reporting system in India is very poor, and without mentioning of cause of mortality, uh, to maintain the statistics of cancer patient data it is very difficult. So it needs to be improvement for the robust tracking of cancer burden. And the Ayushman Bharat digital mission, which has now currently implemented in our country, which describe the registry of health professional, healthcare facilities, uh, ho- all the things they have included in digital mission. But cancer registry, which being the backbone of cancer control program of any country or India also, the national authority did not include cancer registry program in Ayushman Bharat digital mission. So, in conclusion, we found as the digitalization play a crucial role for the functioning of cancer registry, and still in India the registry coverage is very low. Thus, we believe that National Cancer Registry Program should be integrated with Ayushman Bharat Digital Mission to strengthen the process of cancer registration across the country. And there are still many unmet needs and patchwork of public health infrastructure in countries like India. This initiative will aid in the country pursuit of universal health coverage. Thank you. Thank you so much.
Thank you, Dr. Subarajan Jana. That's a wonderful presentation. And does anyone have any query? Hope there are no queries. Thank you, Dr. Subarajan Jana, for your wonderful presentation again.
Thank you so much. Thank you so much.
Yeah, thank you. Okay, we move forward in the webinar, and the next presentation is by Dr. Almeriv Kefe from University of Gondar, Ethiopia. Hello, Dr. Almeriv. Hello, Dr. Almeriv. Are you ready with your presentation? The next presentation is by Dr. Alumarov Kefil from University of Gondar, Ethiopia. Hello, Dr. Alumarov. Are you ready with your presentation? Okay, let's wait for Dr. Almarov for a few more minutes. Hello, Dr. Almarov. Can you please unmute yourself? Yeah. Hello, Dr. Almarov. Hello, Dr. Almarov. Hello, Dr. Almarov. Hello, Dr. Almarov. Are you ready with your presentation? Okay. I think there is a technical issue from Dr. Almarov Kefil, so let's wait for him for a few more minutes. Hello everyone, sorry for the inconvenience caused as we have moved ahead with the schedule. So we take a lunch break and continue remaining sessions after lunch. Stay connected until 30 minutes from now. Thank you.
Thank you.
This conference will now be recorded.
I hope everyone are back with afternoon session. So let's start our first session from Dr. Ambreen Tausif from National University of Medical Sciences, Pakistan. Hello, Dr. Ambreen.
Hello.
Hel-
So,
Hello. Are you ready with your presentation?
just a minute, please. I have to switch off.
Yeah, yeah. There's a lot of.
So, am I audible? Please. Hello.
Hello. Hello.
Am I
Yes.
audible?
Yes. Yes. You are audible. I'll be making you as a presenter. So kindly
Okay.
click on the share button, please. I'll be making you right now.
Right.
Yeah, I made you as a presenter. Kindly click on the share button and continue your presentation.
Right. So, I'm trying to share it, but I think it's not visible. My
No, no,
screen is
you'll
not visible.
get a. Yeah, your screen is not visible yet. Yeah, yeah. No, you
So,
didn't.
it's
You have to share your. Yes, yes. Now it's visible. Kindly make it a full screen.
right. Just a minute.
Yeah.
So, is it fine now?
Yeah, that's perfect. Please move ahead with your presentation.
Right. Thank you so much. So, a very good afternoon to everyone. I am Professor Dr. Ambreen Tosif, and I am heading Physiology Department from CMH Lahore Medical College and Institute of Dentistry from Pakistan. First of all, I would like to thank the organizers of this conference for providing me this opportunity to talk upon a very novel topic, that is role of GTPase activating RAP and GAP domain-like protein 3 and centrosomal protein 290 as an early diagnostic novel biomarkers for ovarian cancer, a proteomic and integrated bioinformatics approach. Ovarian cancer is the among the gynecological malignancies. It is considered to be the deadliest and the utmost devastating malignancy with the highest mortality rate. It is seventh most common malignancy worldwide, whereas it is the third most commonly occurring gynecological cancer. And it also stands among the fifth main death cause from cancers after breast, colorectal, lung, and pancreatic cancers in women. In Pakistan, its incidence is also increasing, and now it stands among the first three female malignancies with an increasing incidence of 13.6%. According to International Federation of Gynecology and Obstetrics, stage 1 ovarian cancer is confined to ovaries. It has been divided into 4 stages, whereas the stage 1 is confined to ovaries, whereas stage 2 ovarian cancer has metastasized to near locations within the pelvic cavity, such as fallopian tubes or bladder. Whereas stage 3 ovarian cancer is diagnosed when it has metastasized to the retroperitoneal lymph nodes or outside of the pelvic cavity. And we call it as stage 4 when it involves malignant cells in pleural effusions and metastasize to distant sites based on the graphic created by Cancer Research of United Kingdom. The dilemma of this disease is associated with its significance, with its identification at the later stages. Owing to the absence of specific signs and symptoms during the initial stage of the disease and the unavailability of early screening and diagnostic tests, Most of the ovarian cancer, that is 79.2%, they have been diagnosed at an advanced stages, that is FIGO stage 3 and 4, with a high mortality rate. Whereas, as explained by International Federation of Gynecology and Obstetrics, if the cancer is detected at an early stage, that is stage 1 and 2, then the 5-year survival rate can be achieved in 90 to 95% cases, whereas it is only 28 and 10% for advanced stages, that is stage 3 and 4 respectively. Hence, the timely diagnosis for ovarian cancer at this stage, at this early stage, is critical for its fundamental control and successful treatment. Currently accepted and widely used serum biomarker, that is cancer antigen CA-125, lacks the sensitivity and specificity for early stage diagnosis, unfortunately. It is non-specific in advanced stages and neither sensitive nor specific in early stage ovarian cancer diagnosis, because it is elevated in approximately 70 to 90% of women with advanced stages and only 50% in early stage of ovarian cancer. Moreover, it is also found to be elevated in other benign and malignant diseases, endometriosis, benign ovarian cysts, uterine fibroids, pelvic inflammatory disease, pregnancy, menstruation, pancreatitis, and liver cirrhosis as well. So, therefore, there is an utmost need to seek an early stage protein biomarkers through a proteomic based approach. Proteomics is a latest approach. It is a state-of-the-art approach to search for specific and sensitive markers for the early diagnosis of ovarian cancer with much improved diagnostic accuracy. What is proteomics? It is basically a comprehensive protein profile expression analysis and identification and its quantification of a whole cell, tissue, organ, or a whole biological system at one point in time. So the objectives for this study were to determine the ovarian tissue proteomic profiles in women with ovarian cancer. Secondly, to compare the proteomic profiles of ovarian cancer with healthy controls, with their healthy counterparts. And whereas the third objective was to validate unique over or underexpressed proteins in ovarian cancer patients. For this purpose, this study was designed as a cross-sectional comparative study, and the place of study where the study was to undertake was School of Biological Sciences, University of Punjab, Lahore, and Department of Physiology and Cell Biology, UHS, Lahore. This study included 40 subjects in total, and we had divided them into 20 each in two groups. Whereas group 1 was consisting of those ovarian cancer women who were aged 40 to 65 years, and whereas group 2 was considered to be the control group, and it was consisted of those women who were scheduled for hysterectomy with bilateral salpingophorectomy for a reason excluding other ovarian cancer. That is the for the procedure other than the ovarian cancer, they have been underwent surgery, and they were of the same age, that is 40 to 65 years. We included all those cases who have been diagnosed as ovarian cancer patients, and based on the clinical history, CT scan, abdominopelvic ultrasound, then transvaginal and abdominopelvic examination and ultrasound, and CA-125 as well as MRI. Whereas the controls were taken for this study, they were those one who were screened negative for ovarian cancer but scheduled for hysterectomy with bilateral salpingophorectomy other than ovarian cancer. Whereas we have excluded those subjects who were pregnant at the time of inclusion, or who were suffered from endometriosis or benign ovarian cyst, or who were diagnosed with diabetes mellitus, malignancies, or hepatitis B, hepatitis C, or who were on chemotherapy. After getting approval from Ethical Review Board of University of Health Sciences, written informed consent was taken from all the participants, and complete demographic information and detailed medical history was recorded on a pre-designed pro forma. We collected 5 milliliter of venous blood sample under aseptic technique, and then the serum was separated by centrifugation at 4000 rpm for 10 minutes at 4 degrees centigrade, and they were further evacuated, labeled, and stored at -80 degrees centigrade till further analyzed by ELISA. The sample which were collected, they were consisted of both malignant and healthy ovarian tissue. And just after excision of excised malignant ovarian tissue, they were snap froze in liquid nitrogen and stored at -80 degrees centigrade. And the similar procedure was undertaken for non-malignant healthy ovarian tissue as well. And the histopathological report for both, that is for malignant as well as for healthy samples, they were obtained for both samples in order to clarify or in order to label them as malignant or healthy ovarian tissue. These are the pictures which were captured while collecting the samples. Here you can see the healthy ovaries, and whereas the ovarian tumor stage 2 ovarian cancer can be seen here, whereas this is a picture taken from ovarian tumor stage 3, and this was the sample which was taken from ovarian malignant tumor, that is stage 4 perioperatively, and this was the resected ovarian tumor just after the resection, and the samples was taken. This flow chart is showing the overall methodology which was conducted throughout the study. That is after sample collection, that is for both ovarian as well as healthy malignant ovarian tissue. Then these samples, the tissue lysis was done and the protein extraction was done, and this protein extraction was then quantified by Bradford assay, and the overall protein measurement was done of both samples, that is healthy and malignant. Then after this, the 1D page analysis was done to in order to have the snapshot of the overall protein content present in all samples. And then after this 1D STS page, this was followed by two-dimensional gel electrophoresis, that is 2D electrophoresis analysis. And then these 2D gel images from taken from all the samples, that is from healthy as well as for malignant ovarian tissues, these were analyzed by using a software which is known as a Syngene dimension software. And after the analysis by this Syngene 2D analysis, the all those spots which were either overexpressed or underexpressed seen in all gels uniformly, they were statistically analyzed. And then those gel spots who have shown the 1.4 change difference, volume difference, fold difference between as compared to healthy gels, these spots were excised, and then these were identified and picked up from these 2D gel protein spots. These protein spots, which were actually differentially expressed in all gels of malignant ovarian cancer gels as compared to their healthy counterparts, they were further underwent for tryptic digestion. And these protein spots, after passing through this tryptic digestion, they have been converted into peptides, small peptides. And then after this, these small peptides, they were analyzed and examined by multi-TOF mass spectrometry, that is matrix assisted laser desorption and ionization time of flight mass spectrometry analysis. And from this mass spectroanalysis, they were identified as a peak masses or the peak waves, which spectra in the form of a spectra, and these spectra, they were further explored by using different databases like the NCBI and Swiss Prot databases. And by with the help of these databases, these were meshed against the already provided data bank for all the proteins, that is over 1800 million proteins present in this data set. And for further validation, they were passed through the in silico analysis, that is the bioinformatics tool. And these are the computational tools such as String and Panther. And after passing this, they were finally validated by using ELISA. And this ELISA measurement would give rise to the candidate protein markers that were novel in this study, and they have been identified before. And all those novel identified protein potential biomarkers, they have been further passed through the recessive operative curve and to determine their sensitivity and specificity in order to discriminate the healthy patients from the malignant ones. And at the end, data interpretation and statistical analysis was done. Statistical tests which were used during this study was, that is, the data analysis was done by using SPSS version 25. Then for the determination of volume of each differential gel spot showing one-fold change, increase or decrease in the change of expression, student t-test was applied, and then mean and standard deviation was taken, and positive protein identification confidence limit was taken as with the confidence interval of 95% with a p-value less than 0.05. Several build test was run to check the normality of data, and then one-way ANOVA of candidate biomarker was performed. And those proteins who have shown the statistically significant difference, they were passed through the post hoc analysis. They were further analyzed by Tukey's test to execute multiple comparison between all stages of ovarian cancer in healthy women, because we have taken samples from stage 2, stage 3 and stage 4 ovarian cancer, and they were compared against their healthy counterparts. At the end, for the determination of sensitivity and specificity and to compare the diagnostic performance of these potential identified novel biomarkers for the early detection of ovarian cancer, ROC, that is recessive operative curve, was plotted. And the graph pad prism 8.2.1 was employed for statistical analysis of ELISA data. These were the results which were obtained after having all this methodology. That is, first of all, after the tissue lysis, the protein quantification, after protein quantification by Bradford assay and their quantification, they were the first, the 1D STS gel was run, and this is the picture showing different bands, and with the marked in the red arrows which show the overexpression bands in the tumor tissues. Here you see these are all the malignant tissue representing by T1 to T5, whereas H represents the healthy ovarian tissue. So these were compared with their healthy samples. So this shows the overexpressed bands as compared to their healthy counterparts, and whereas underexpressed proteins were also there. So this picture shows the overall snapshot or the overall picture of the protein present in all samples. Then they were further passed through after 1D analysis. These samples, they were further analyzed by using the IEF, that is isoelectric focusing, and then they were run through the two-dimensional gel electrophoresis in order to separate all the proteins according to their molecular weight and their ionization point. So this is the picture which has been taken from normal or the healthy ovarian tissue, and here you can see there are lots of protein spots which were obtained after passing through the all these stages. And this is the picture, one of the representative gel, 2D gel, taken from the stage 2 ovarian cancer tissues. And this is the representative gel showing multiple protein spots presentation and representing the ovarian cancer stage 3, and here is ovarian cancer stage 4 representing multiple protein spots. These are the 2D gels which are taken from these respective ovarian cancer patients. And after having these 2D gels, they were further analyzed, as I told you before, that by a software which is known as Dimension Syngene software. And these protein gel spots from the healthy ovarian tissue, here you can see these are the gels from the healthy ovarian tissue, and all these gel spots detected by the Dimension Syngene automatically, whereas these are the spots which have been detected by manually. These are all the differentially expressed spots, because these spots are either overexpressed or underexpressed, present in all gels from healthy as well as from ovarian cancer tissue. So these protein gel spots, these are the analysis of dimension syngene analysis, software analysis, and these are the pictures taken from ovarian cancer tissue. Again, the same gel spots detected by dimension syngene itself, and these are the manually spotted gels spots. This is the comparative analysis of healthy ovarian tissue, all possible detected spots and then manually detected spots. So after having these dimension analysis and these statistically gel spots, these gel spots which show the statistical difference and they were found to be statistically significant, they were further picked up. and they were passed through tryptic digestion. And after passing through the tryptic digestion, they have been converted into peptides, and these peptides, they were further analyzed by multi-TOF MS analysis. And with the help of multi-TOF analysis, total we have profiled 112 proteins identified in ovarian cancer in all samples, which were commonly present in all samples. And these are those 112 proteins which were initially profiled. They were further showing a significant mascot score. Mascot is a search engine which has a definite score, which should be, the cutoff value is 56. So all those protein spots among these 112 who showed the significant mascot score, they have been further isolated. And out of these, we got 44 differential protein identified in ovarian cancer. All those 44 which showed the significant mascot score, that is more than 56. Then after from these 44 differential proteins, we had further shortlisting them on the basis of 4-tier criteria, that is the peptide mass fingerprinting score. That is, it should be those proteins who showed more than 79 pmf score, p value should was less than 0.05, and false discovery rate was less than 0.05. All those proteins out of these 44 who fulfilled this 4-tier criteria, these were found to be 31. And these 31 proteins, they were further identified with 95% accuracy confidence, because they have fulfilled this 4-tier criteria. And these 31 proteins, out of these, 9 were overexpressed proteins which were found to be present in all gels of ovarian cancer, and 22 were found to be underexpressed as compared to healthy gel. And out of this further shortlisting, 9 overexpressed and out of 22 underexpressed, we found 6 novel proteins, 3 overexpressed and 3 underexpressed proteins which were identified. And out of these 6, only 3 novel proteins, they have been validated by ELISA in order to determine them, or in order to nominate them as a potential biomarker for the early diagnosis of ovarian cancer. So here you can see, this is a picture of 2D gel proteomic profile of malignant ovarian tissue taken from stage 3 ovarian cancer patient. And here you can see all these 112 proteins which were profiled, which I just mentioned here, that is total 112 proteins were identified in ovarian cancer initially. So out of these initially profiled 112 proteins, then we will further shortlisted them up to 6 novel proteins. And this is the picture taken from healthy ovarian tissue 2D gel, a representative picture. And this is the 2D gel picture from malignant ovarian tissue 2D gel. And here you can see all those protein spots which are marked and encircled as red. They are basically overexpressed in malignant ovarian tissue as compared to healthy ovarian tissue 2D gel. Whereas all those spots which are encircled as yellow color, they are underexpressed protein spots, which were found in all malignant tissue ovarian tissue gels as compared to their healthy counterparts. So these were numbered, profiled, and further shortlisting lead them to this. So this is the overall picture. That is the after this whole process is known as peptide mass fingerprinting (PMF) score. And first of all, they with the help of this technique, we identified 112, and out of this 112, significant proteins were found to be 44 by searching against the mascot data search base and Swiss broad database. And then ultimately we found to have the 9 overexpressed and 21 underexpressed proteins. So this is the list table showing overexpressed proteins. There are 9 overexpressed proteins which fulfilled the 4-tier criteria and found to be present in our study. But here all those proteins which are mentioned in black color, these are those proteins which have already been identified in different studies. So the those proteins, that is the protein spot number 28, that is centrosomal protein of 290 kDa, and 101 spot number, that is GTPase activating RAP-RAN GAP domain like protein 3. These are our potential candidate protein as well. So these two proteins which were found to be overexpressed, they are totally novel proteins, and they have not been discovered before in the world in ovarian cancer. And these are the proteins, 21 proteins, which were underexpressed in nature and found to be present in malignant tissue 2D gels as compared to healthy 2D gels. And among all these proteins, only the one protein was found to be a novel, that is actin related protein 3, which was further validated by ELISA. So this is the peptide mass fingerprinting score of one of the our candidate proteins, that is GTPase activating rep and GAP domain like protein 3, that is GRNL3 protein. And this is the mass spectra and the waves. And this is the total protein score, mascot score, which has been obtained by searching against the mascot engine. And this is the sequence coverage of that protein among that database. And this is the same peptide mass fingerprinting wavelengths and the mass spectra and the score, significant score, and the total peptide sequence coverage. This is the for to further validate and to further in silico characterization of these MS identified protein was performed by using bioinformatics tool, gene ontology and protein analysis through evolutionary relationship, that is Panther databases, and by using search tool for the retrieval of interacting genes and proteins, that is string, to reveal functional interactions among all identified dataset proteins. The protein of interest in ovarian cancer was GARNL3. That is, here you can see this is the protein GRNL3, and it shows the different functional interactions with the other proteins present in the data set, or these are those proteins which have been involved, which appear to be interacted with those proteins that are directly involved in cellular proliferation, tumorigenesis, apoptosis, inflammation, and immunomodulatory processes for other cancers occurring in our body. So this is the protein, that is the candidate protein, second candidate protein, that is the CEP290 and centrosomal protein, and it is also showing the different functional interaction with the other proteins which have been involved in tumorigenesis or other malignant pathways by mutations or by other immunomodulatory mechanisms. And the second bioinformatics tool in integration was done by functional and structural classification of identified ovarian cancer protein by protein analysis through evolutionary relationship, that is Panther. And it has been shown that these two proteins, our candidate biomarkers, have been involved in biological processes as well as in different cellular processes which have been involved in in the pathogenesis of ovarian cancer, as well as in metabolic processes. So these proteins, they were found to be involved in the pathogenesis of ovarian cancer. So after this, all this computational bioinformatics, GARNL3 serum quantification was done in healthy as well as in ovarian cancer women by ELISA. And here you can see that the ovarian cancer, when quantification between healthy and ovarian cancer was done, then the levels were found to be 4 times higher, that is 58.29 in ovarian cancer as compared to their healthy counterparts, that was only 15.46 plus minus 6.81. And similarly, quantification was also done among all stages, and you can see that stage 4 was found to be showing the highest level of GRNL3 as compared to stage 3 and stage 2, and as well as when compared with their healthy patients, levels found in the healthy patients. And similarly, CEP290, the second biomarker for the early detection of ovarian cancer, the serum quantification in healthy and ovarian cancer women by ELISA was done, and the levels were found to be again 4 times higher in malignant ovarian cancer as compared to their healthy women. And the quantification was also done among all stages of between stage 4, stage 3, and stage 2, and this shows the statistically significant difference between all stages as compared to their healthy women levels of CAP290. So this is the p-value showing the GRN3 and CAP290 showing the significant p-value. Here you can see the post hoc Toki's test was done to execute the multiple comparison between all stages, that is from stage 2 to stage 4 of ovarian cancer, and compared with the healthy subjects, and it was found to be statistically significant. Stage-wise serum levels of GARN3 and CP290 was done, and it was found to be statistically significant. Similarly for CEP290 as well. So it shows the p-value with the statistically significant difference. So here comes the ROC, or the analysis by receiver operating characteristic curve. That is this is the curve which was plotted, or which was determined in order to discriminate between the healthy patients as well as from the malignant ovarian cancer, all those patients who will be suffering from malignant cancer. So this was found to be with the sensitivity 94.4% and specificity was 90.9%, and the cutoff value for GARNL3, if it was found in malignant cancer patients, that it will be 28 nanogram per milliliter. It is the cutoff point which can discriminate, or which can discriminate between the healthy as well as the ovarian cancer patients. And with the area under curve was found to be 0.987 with the statistical p-value. And similarly for the other biomarker, that is CEP290, again the sensitivity was found to be 92.72% and specificity was 88.57% with the cutoff value of 21 nanogram per milliliter to discriminate between healthy and ovarian cancer suffering patients. With the area under curve was again found to be statistically significant, that is 0.95. So, with the help of all this, we can conclude that these two biomarkers, potential biomarkers, or the candidate biomarkers for the early detection of ovarian cancer, they can be used as a minimally invasive diagnostic biomarker for the early stage disease, which will can produce less financial burden of chemotherapy and healthcare, and definitely it is going to improve the healthcare cost for all those poor patients who cannot bear all the expenses of this expensive treatment. It can also be used as a screening tool in high risk group, or especially in those groups who have positive family history having ovarian or the breast cancer. So we can use these biomarkers in those groups in order to identify at an early stages. And it can also use as a prognostic marker. It can show the, or it can determine the disease progression as well as the treatment progression, and it can show the efficacy of treatment as well. So with the help of this, we can possibly detect those ovarian cancer patients in high risk groups with a positive family history of breast and ovarian cancer. We conclude that this study identified an upregulated expression of two novel biomarker proteins, that is GARNL3 and CEP290, as a potential biomarker, and it can provide insight into the mechanism and the pathogenesis of ovarian cancer for its prognosis and early detection. And in order, it will reduce the mortality as well as the morbidity. This is the bibliography used. Thank you so much for your attention.
Thank you, Dr. Ramran. That was a wonderful presentation. So, anyone have any
Thank you so
query?
much.
Okay, hope there are no queries. That was a wonderful presentation by Dr.
Indeed.
Amreen Tausif.
Thank you
And
so
let's move
much.
further. Thank you.
Thank
Let's
you.
move
Thank
further
you.
with the yeah. Let's move further with the session. The next presentation is a video presentation. It's a video presentation by Dr. Leila Mahmoud Montasser from Menofia University, Egypt. So I'll be yeah. It's a keynote presentation by Dr. Leila Mahmoud Montasser. So I'll will be sharing the video from our end.
I am Dr. Leila Mahmoud Muntasser, Professor of Clinical Pathology, Faculty of Medicine, Manufaa University, Egypt. The title of my presentation is 3D bioprinted stem cells for lung regeneration. Distinguished Professor of Clinical Pathology and Hematology, Chief Scientist, Chair, Stem Cell Regenerative Medicine, Nanotechnology and Tissue Engineering, SRNT Research Group, inaugural holder of the CERN Chair, Chair Emeritus, founder leader of Clinical Pathology Department, Faculty of Medicine, Manufaa University, Egypt. Muntasser is a global award-winning and a champion of stem cell technology and nanomedicine for more than 1 decade. In the era of COVID-19, she was awarded 44 certificates of appreciation for successfully presenting 53 global webinars for outstanding contribution. The certificates were presented in recognition of an outstanding university professor for her devoted and untiring efforts to educate scientific society and her students and public awareness on the COVID-19 pandemic, for her ideal command in leadership her university to achieve their role in society, for her relentless modality to push a quality tactic and culture to act, and for doing as role model to all. Manufaa University is located in the beautiful city of Shibin el-Kom in the south of the Nile Delta and overlooks one of the main branches of the Great Nile River. The upper photos front view, while the lower two photos are back view. Outline. I will start with summary of my presentation, then brevity design on some of my experience in the era of COVID-19, then synopsis draws on some of my deep expertise in the domain of regenerative medicine, and lastly, conclusion. Summary of my keynote presentation at Second European Congress on Tissue Science, Stem Cell and Regenerative Medicine, 2023, June 29 to 30, Barcelona, Spain, titled 3D bioprinted stem cells for lung regeneration. Coronavirus stormed the lungs in human beings, reproducing constant deterioration. Contemporary obtainable cures cease in heavy illness, whereas various retargeted antiviral and host response medications are under exam as possible therapies. Other new therapeutics are also being searched to relieve the impacts on serious sick cases. The harness of stem cells has been idolized as a vital agent for humans' tissue innovation. But existing technologies lacking the power to create plentiful tissue engineered styles with the energetic identity of the basic tissue. Objective is to replace and modernize deteriorated lung tissue by tissue engineering. The utilization of mesenchymal stem cells for COVID-19 could become a fascinating path. The structural and functional complexity of the pulmonary system makes it well suited to 3D printing technology. The 3D bioprinting technology of stem cells harnesses the union of the bioink that intends as a living framework and several modalities of cells with the entity of alchemical agents and biomolecules to found an intense and workable in situ 3D functional construction. The modern evolution of 3D printing technology allows the scopes of enlarging biopure multiplex tissue communications by modernization of cells, biomaterials, and bioactive accumulations in theranos. Progress in 3D printing technology enables the prospects of originating functional human tissues in the laboratory, looking to evidence tectonic and effective uniformities as mother tissue in the human structure, besides to push guided tissue evolution such as segregation of stem cells. We look at modern strategies to upgrade cases living opposition to coronavirus using stem cells and its pharmaceutical advantages with applying 3D printing technology. With that rapidity of development of science and clinical advance in the harness of 3D by printed stem cells as a cell treatment of serious pneumonia, here we may revealing a novel epoch of coronavirus treatment. Briefly, 3D printing starts with modeling, where visual blueprints from computer-aided design. Then the second step is printing, wherever 3D printers read the design and lay down successive layers of materials. And lastly, finishing, with what after printing, the supports are removed or dissolved to get the final product. 3D bioprinting integrates the conventional 3D printing, imaging, and cell gel to fabricate functional tissue for regenerative medicine, pharmaceutical, preclinical drug screening, and animal-free meat. Current tissue engineering strategies lack the capability of fabrication of fully functional tissues and recapitulate their heterocellular structure. 3D printing shares the three basic components with the conventional Gutenberg paper printing. The 3D model file to be printed, blueprint, is analogous to the text file. The bioink, which comprises cells and other bioactive materials, is analogous to the ink, and the 3D printer is analogous to the printers that deposit the ink on a substrate or print platform. Bioprinting has a great potential to surpass the traditional tissue engineering techniques and can offer solutions to many existing technological hurdles. Overview schematic of the bioprinting processes. Bioprinting starts with obtaining the anatomical structure of the target tissue by a proper imaging technique such as CT and MRI. Specialized software is then used to translate the image to computer-aided designs, drawing of cross-sectional layers, such that the printing device will be able to add them in a layer by layer process. Next, the bioprinting device constructs the tissue using a specific printing method, such as inkjet 3D bioprinting, micro-extrusion 3D bioprinting, laser-assisted 3D bioprinting, and stereolithography by employing a combination of printing materials such as scaffold, bioink, and other additive factors. The accuracy, stability, and tissue viability vary through these processes. Finally, the constructed tissue is post-processed in a bioreactor to recreate the required in vivo environment to maintain tissue viability during the maturation period. Brief summary of my presentation at Technology, Industry, People and Economics Conference 2023, January 24 to 26, New York, USA, titled The Latest Technology in 3D Bioprinting. Present technology is incapable to make copy tissue engineered structures with the equivalence of the natural tissue. The modern development of 3D printing technology enables the fabrication of tissue substitutes integrating cells, biomaterials, and bioactive compounds, and the concept of in situ 3D bioprinting in the lab to cut out the middle step of in vitro growing cells and implanting cells directly into the body to repair damaged tissue or organ via tissue engineering. This type of technology is less invasive, offers great advantages to the patients, and hopes to offer a new solution for those on the waiting list. Summary of my keynote speech at 5th International Conference on 3D Printing and Additive Manufacturing 2022, August 22 to 23, Singapore City, Singapore, titled Robotic In Situ Bioprinting Technology for the Generation of Functional Engineered Tissues. Bioprinting has provided several advantages to traditional tissue engineering approaches for fabricating scaffolds for organ tissue regeneration. Nevertheless, this technology, also known as in vitro bioprinting, suffers from several limitations when considering its clinical application, such as scaffold handling difficulty, risk of contamination, need of maturation period in a bioreactor, and shape morphology of the bioprinted construct not perfectly matching with the defect site. For these reasons, in situ bioprinting has emerged as a promising alternative technology. The aim is to report robotic-assisted in situ 3D bioprinting technology for tissue regeneration. In situ bioprinting consists in the direct deposition of biological material into the patient, following the complex geometry of the anatomical defect to achieve desirable mechanical properties suitable for tissue regeneration. This approach guarantees an enhancement in the maturation and differentiation of the bioprinted constructs, since the patient body itself works as a bioreactor. 3D bioprinting could be a promising technique for the generation of functional engineered tissues. By 3D scanning the shape of the Damaged area can be easily assessed. Then, the shape of the 3D printing object is determined with reverse engineering software in a few minutes. This technology makes it possible to repair defects with complex shapes in a short time. Advances in robotic surgery fuse imaging and computer-assisted medical interventions could be integrated to develop future clinical in situ 3D by printing processes, which can be translated to products for a variety of surgical applications. Printing the devices directly onto organs in a minimally invasive manner could help to reduce the need for implant surgeries as long as there is a seamless integration between the printed material and the biological matter inside the body. Interoperable three-dimensional fabrication of living tissues could be the next biomedical revolution in patient treatment. Brief summary of my keynote speech at Annual Technology, Industry, People, and Economics 3D Printing Conference Virtual Global Event 2022, January 18th to 20th, titled Modern Technologies. Where are we living? 3D printing is all about cutting out the middleman. This benefit is being carried over to bioprinting in the laboratory to cut out the middle step growing cells in the lab, in vitro, and just implanting stem cells directly into the body for growth in situ. In situ bioprinting could be used with robotics for some surgical procedures as it is less patient invasive with less pain, less recovery time, less chance of infection, and less time spent in the hospital. Anticipate the utilization of mobile printers for emergency cases and in developing countries and distant zones. 3D bioprinting is an additive biomanufacturing technology having potential to fast forward translational research, as it had the capability to fabricate artificial tissues and organs that closely mimic biological tissues or organs. As an emerging area of research in the field of tissue engineering, 3D bioprinting has scope in the development of implantable tissues and organs, construction of tissue organ models, and high-throughput diseased cancer models for pharmaceutical and toxicological studies. Further, this area has diversified with the continuous upgrading of 3D bioprinters and biomaterials, which play major roles in the architectural quality and functionality of bioprinted construction. Addressing these technological complexities requires an integrated approach involving expertise from different areas of science and engineering with lateral thinking. Bioprinting remains a promising solution for addressing the increasing organ shortage for transplantation globally. The ability to generate tissues for transplant with a lower immune response risk holds significant promise in the fabrication of artificial organs. The recent progress in hydrogel science, including the development of dynamic switchable hydrogels and oxygen-producing hydrogels, provides more and more methods that enable control of cell microenvironments. However, the full potential of 3D bioprinting can be realized by improving upon the printing speed, capability to bioprint at various scales, availability of bioprintable materials and hydrogels, vascularization of tissues, innervation of tissues, and on-demand skillful fabrication and cell maturation mechanisms. 3D bioprinting involves printing cells and materials like extracellular matrix components together to form a viable tissue construct following a one-step biofabrication process. This is followed by incubation of the 3D printed tissue or organ in a bioreactor before any further experimental procedures are performed, and then passed on for preclinical and clinical trials. Summary of my keynote speech at 3D Printing and Additive Manufacturing 2022, April 13 to 14, titled In situ 3D Bioprinting for Tissue Engineering. Tissue defects resulting from disease, trauma, or surgical resection require treatment to restore tissue structure and function. There are certain challenges related to reconstructing complex tissue defects. Such defects are difficult to treat using conventional and standard size implants. They need to be treated by using modified or trimmed implants or constructs. The implementation of printed tissues also adds complexities due to the difficulty in maintaining the structural integrity of fabricated constructs. To tackle these challenges, the concept of in situ 3D bioprinting has been suggested. Objective is to fabricate grafts in vitro, then be implanted in vivo. In situ 3D bioprinting, in which tissues are directly printed at the site of injury or defect, combined with cells freshly isolated from patients to produce custom-made grafts that resemble target tissue and fit precisely to target defects. The constructs are designed and engineered ex vivo and subsequently implanted into the body. This novel research field provides great potential for tissue engineering and regenerative medicine applications where pre-operative planning of construct size and shape is difficult or impossible to predict. 3D printing could provide constructs that can be pre-designed to precisely fit defect size and shape. This ideal solution would employ a commonly used 3D printing technique in the operating room and 3D print constructs in situ. When fully deployed, the technology of in situ 3D printing will result in more accurate reconstruction of tissue defects and result in faster and more efficient healing of tissue defects. 3D bioprinting is an emerging biofabrication technology, driving many innovations and opening new avenues in regenerative therapeutics. Advances in robotic surgery, fused imaging, and computer-assisted medical interventions should be integrated to develop future clinical in situ 3D bioprinting processes, which can be translated to products for a variety of surgical applications. Isolation protocol of mesenchymal stem cells, mononuclear cell separation from bone marrow on fecal, primary culture of mononuclear cells, then subculture of isolated mesenchymal stem cells. The cells from primary culture were seeded for first and second passage. The left figure shows electrospinning set. The middle figure shows diagram of electrospinning setup, while the right one shows scanning electron microscope image of a poly-L lactic acid scaffold. Conclusion: bioprinting offers tremendous potential for the area of tissue regeneration and repair. 3D printing is emerging as a powerful tool for tissue engineering by enabling biofabrication technology, driving many innovations, and opening new avenues in regenerative therapeutics. The aim of 3D bioprinting is to fabricate grafts in vitro, which can then be implanted in vivo. This technicality gives the podium for innovation of organs and tissues in a tidy style. 3D bioprinting is a rapidly evolving field of research with immense challenge but tremendous potential to revolutionize modern medicine and healthcare. Thank you.
So that is a keynote presentation. Okay, that is a keynote presentation by Dr. Leila Mahmood Montasser. For any queries regarding the presentation, kindly reach our program manager. She will guide you accordingly. So by moving further, the next is the video presentation by Dr. Alexander Markin from FSBSI Petrovsky NRCS Russia. So we'll be playing the video presentation from our end.
Hello, dear colleagues. My name is Alexander Markin. I'm a senior researcher at the Laboratory of Cellular and Molecular Pathology of the Cardiovascular System, which is part of Petrovsky National Research Center of Surgery. Today, I would like to present to you a part of our project. The scientific group under my leadership deals with the following issues: extended characterization of the cellular composition of the intima in normal, pathological, and experimental conditions. Next field of research is the development of approach to the creation of cellular models of atherosclerosis for its early diagnosis and therapy. And the last topic, the development of a method for mitochondria-mediated anti-atherosclerotic therapy. But today I want to stay on the first and the second topic. The main theme of our laboratories group is atherosclerosis. It is a chronic immunoinflammatory disease of the aorta and large arteries. Atherosclerosis underlies the development of various severe and life-threatening complications, including such fairly common causes of death and disability as occlusion or obstruction of the arteries. A huge number of recent studies have not revealed an unambiguous factors provoking the development of atherosclerotic lesions. Among a large number of players, we can single out the main ones, namely a pathological change in the population composition of intima cells, a violation of transendothelial transport, a change in lipid metabolism with subsequent accumulation of cholesterol by phagocytic cells and the formation of foam cells. Currently, there is a stable thesis that inflammation and increased oxidative stress are the main causes of atherosclerosis. Plasma low-density lipoprotein can penetrate through the artery wall and then undergo various modifications. Multiple modified low-density lipoprotein is a powerful inducer of inflammation, thus lowering plasma LDL levels and suppressing inflammation have become the main focus of atherosclerosis treatment. In addition, multiply modified low-density lipoprotein contribute not only to the disruption of lipid utilization processes, but also to the modulation of changes in the composition of intima cells. The process shifts the balance of cellular interaction towards the pro-inflammatory activation of resident and circulating immune cells. This provokes a local immunological reactions leading to uncontrolled uptake of low-density lipoprotein by macrophage-like cells through scavenger receptors or by efferocytosis. These processes generate a vicious circle of immunological and metabolic disorders at the cellular level. This leads to the formation of a large number of foam cells and the subsequent formation of a necrotic nucleus of an atherosclerotic plaque. However, speaking about the determined role of immune cells, it should be noted that all types of intima cells have the ability to uptake of excess LDL lipids. These can be smooth muscle cells, pericyte-like cells, and even endothelial cells. The study of the mechanisms leading to the formation of foam cells will reveal the key factors that trigger the process of atherogenesis. In particular, activation and induction of the phenotype of foam cells in some endothelial intima cells will help to understand the basic principles of initiation and development of atherosclerosis. In the course of our work, 18 aortic samples were obtained. The material was obtained after surgical intervention during the aortic plastic surgery from patients of Petrovsky National Research Center of Surgery with their voluntary informed consent. When working with tissue, the samples were treated with proteolytic enzymes. Um, the um, the samples was divided into three layers: intima, media, and adventitia. Dissociation of this tissue is carried out of 3 Petri dishes for each layer of the aorta separately in the volume of cocktail of enzymes. The optimal incubation protocol include collagenase 2 and collagenase 4. Layer by layer tissue dissolution was used, namely the layer of interest was dissolved in a cocktail of enzyme for a fixed time, after which the isolated cells were washed and transferred to culture plate or immediately transported to laboratory for fluorescent activating cell sorting. At the moment, we have started a series of experiments to determine the cellular composition of intima in pathology. To do this, we use the following set of markers for immunophenotype in cells. CD3-1 antigen or platelet endothelial cell adhesion molecule 1, PKM1. It's expressed on surface of myeloid germ stem cells, on the surface of platelets and endothelial cells. The CD34 antigen, CD35 antigen was present on the surface of all human leukocytes, erythrocytes, lymphocytes, eosinophils, monocytes, basophils, and neutrophils, and endothelial cells. CD56 antigen is an isoform of a nerve cell adhesion molecule NCAM. These markers expressed on the surface of some subpopulation large granular lymphocytes, skeletal muscle cells, and of sometimes in hematopoietic stem cells. The CD146 antigen is constitutively expressed on the endothelium and is also found on the surface of other types of cells such as smooth muscle cells and pericytes and other cells. As for peripheral blood leukocytes, CD146 is expressed only on the surface of subpopulation of activated T lymphocytes. CD73 is expressed on the surface of mesenchymal stromal cells, T and B cells, dendritic cells, epitheliocytes, and endotheliocytes. CD105 antigen is expressed mainly on the surface of mesenchymal stromal cells, endothelial cells, and to a high degree in pre-B lymphocytes and a subpopulation of monocytes. The CD13 is found on the surface of mesenchymal stromal cells, most cells of myeloid origin, including neutrophils, eosinophils, basophils, and monocytes. Next markers is human melanoma-associated chondroitin sulfate proteoglycan, or NG2. It's a glycoprotein. It's not found on the surface of normal hematopoietic cells, but is expressed by a subpopulation of myeloid and lymphoid cells with chromosomal abnormality. And it's also expressed on the surface of human glial cells in normal condition. Next markers is PDGFRB, platelet-derived growth factor receptor beta, or CD140B, a protein from the family of receptor tyrosine kinases. It's expressed on the surface of mesenchymal stromal cells. On the presented slide, you can see an example of our strategy for gating newly isolated aortic intima cells. At the moment, we were interested in cells with the pericyte immunophenotype, CD45 minus, CD31 minus, CD34 minus, CD56 minus, CD146 plus, CD73 plus, CD105 plus, CD18 plus, CD13 plus, NG2 plus, and CD140B plus. So in freshly extracted non-patient tissue, the number of such cells is naturally small. Only about 5% of cells do not carry markers of hematopoietic and endothelial cells as CD45, CD31, CD34, and CD36 markers. And these cells was positive for pericytic markers, CD146, NG2, and PDGFRB+. In addition, these cells have markers of mesenchymal stromal cells as CD73, CD105, and CD13. At the same time, these cells changes after the first passage, so we can observe what the percentage of cells that lack next markers, CD45, CD31, CD34, CD56 grows to 55 or 60%, and 80% of these cells carry parasitic markers, CD1, 4, 6, and G2 and PDGFR-beta. In addition, the patient cells were transferred to confocal cups a day before confocal dishes a day before staining. Then, according to the standard procedure, staining was performed on the first day with primary antibodies to the human markers alpha smooth muscle actin, desmin, and the second day with anti-specific antibodies conjugated with fluorescent labels. It was anti-mouse feeds. Cells expressed all the illicit markers were found in the samples. The second part of our work was to evaluate the phagocytic capacity of isolated and cultured cells of the subendothelial layers of the aorta, using feeds labeled latex balloons, phosphorus carboxylate, produced by in vitro gene. Preliminary results have been obtained for this part of the work. The ability to phagocytosis was assessed by counting the numbers of balls per cell, as well as by measuring the fluorescent intensity of the feed signal by flow cytometry. From comparing the degree of phagocytosis by cells of the subendothelial layer of the aorta, it clearly follows that they are in no way inferior to professional phagocytes. However, additional research is required here to form a more representative group. Several experiments were also conducted to measure the ability of subendothelial cells to accumulate cholesterol during incubation with atherogenic LDL isolated from patients with atherosclerosis. In all experiments, significant differences were obtained between the control group of cells of the subendothelial layer and the group of cells incubated at the presence of low-density lipoprotein for 24 hours. Similar experiments were also carried out with a commercial culture of immortalized parasites and with THP-1 cells. The latter acted as a control of the ability to accumulate cholesterol, since this cell line is one of the main model objects for assessing the ability of cells to capture LDL particles. According to the preliminary data, subendothelial cells have the ability to absorb LDL particles, which leads to increase in the concentration of cholesterol in the measured samples. Moreover, this ability is approximately on the same level with similar abilities of linear parasites and THP-1 cells. The second direction of our research was experimental work with monocytes and macrophages. After entering the bloodstream, monocytes circulate in the bloodstream for 1 or 2 days, after which they settle in tissues and become resident macrophages. Macrophages are found in almost all tissues. They play a crucial role in non-specific defense, innate immunity, and also help initiate specific defense mechanism, adaptive immunity, by attracting other immune cells such as lymphocytes. For example, they are important antigen presenting for T cells. It's a well-known fact that as a result of the influence of certain factors, macrophages are capable of polarization, which allows them to perform their functions at the request of the host organism. But in some pathological conditions, this process is disrupted, which leads either to the development of chronic inflammation, as in atherosclerosis, or to the onset of oncological diseases, with a shift in the balance towards the anti-inflammatory phenotype. As researchers of atherosclerosis, we are naturally interested in the first part of the process, namely the development of chronic inflammation, which in turn is one of the reasons for the formation of foam cells in the subendothelial layer of elastic-type blood vessels. So the first part of our work was to study the ability of circulating monocytes to differentiate into macrophages and accumulate lipids under the influence of various cytokines during joint cultivation. One study group included negative control cells without interleukin and LDL, positive control cells with LDL, and the comparison group cells with interleukins and LDL, and cells containing only interleukins. Lipid accumulations was assessed by the enzymatic method by measuring the content of cholesterol in cells. In this project, we used several cytokines, interleukin 6, 8, and 4. Analysis of the data obtained showed that the pro-inflammatory cytokines interleukin 6 and interleukin 8 affect the increase in cholesterol in macrophages, both in a medium that does not contain LDL and in medium containing LDL. Thus, it can be assumed that pro-inflammatory effectors are possibly capable of inducing the accumulation of cholesterol in macrophages and promoting the formation of foam cells in the artery wall during atherogenesis. It's also interesting that stimulation of cells with the anti-inflammatory cytokine interleukin 4 also increases cholesterol levels, which may indicate a role for this interleukin in cholesterol metabolism. In our next work, we used interleukin 7. This interleukin caused a decrease in cholesterol content in cells while simultaneously incubated with LDL. In addition, analysis of expression of interleukin-1, B, and TNF genes, the basic inflammation mediators, showed that there was no increase of their expression. It's also seen that there is no accumulation of cholesterol in the cell culture. In this way, the addition of interleukin-7 to the cell medium, where LDL was also added. In cells, there is a significant decrease in cholesterol content compared with positive control. Thus, the interleukin-7 may be in some kind of anti-atherosclerotic effect. Further studies should contribute to determination of factors affecting the development of atherosclerosis. Thus, the primary pathological change in cholesterol metabolism lead to an increase in cholesterol content in cells and the formation of foam cells. It does chronic aseptic inflammation lead to a change in lipid metabolism in cells? The answer to this question will allow for our further research. In conclusion, it would like to state that the joint efforts of medical professionals and researchers of biological and biomedical profiles that allow us to correctly solve the fundamental task of combating atherosclerosis. Using clinical data in combination with biological methods will allow us to look at the issue from different angles. The problem of the development of atherosclerosis is only part of a huge list of unresolved tasks of modern biomedical science But even a small progress made in these directions will improve the quality of life in hundreds of thousands of citizens around the world. Thank you for your attention, and please, if you have any questions, send them to me at my email. Thank you.
Well, that is a video presentation by Dr. Alexander Markins. For any queries regarding the presentation, kindly reach our program manager. She will guide you accordingly. The next presentation is by Dr. Braden Luke World from University of Florida, USA. This is a video presentation, so we'll be playing the video presentation from our end.
Remedial. A little bit. Hello, I'm Dr. Lucky. We'll be looking at characterization and treatment of spinal tumors. A little bit of background. Spinal tumors are overall all pretty rare and compared to brain tumors, but can present serious clinical challenges. Most common presentation is due to compression of the cord itself, associated motor and sensory deficits. Primary tumors are characterized by their location, whether intramedullary, so within the cord itself, intradural, extramedullary, cause compression, or primary bone tumors. Back pain is a very common presentation, but they can also have radicular pain based on the location of the tumor and also which nerve roots are being compressed if it's more peripheral. MRI is a absolute standard for terms of detection of the intradural, extramedullary, and intramedullary tumors. Plain radiographs are important too, especially for bone tumors. And overall, the intralesional resection can help guide overall outcomes. A lot of these patients will require adjuvant radiation and chemotherapy, especially for metastasis for the bone, having that ideal margin to help with the radiation control is ideal. But we'll dive into some of the details. So melanomas are pretty common. They can cause progression over time as they grow, even though they're benign tumors. Surgery is typically the option. Most of these are low grade, so don't require adjuvant treatment. Schwannomas, similarly, are extramedullary, can cause some compression as well, can travel out that nerve sheath, and surgery is also curative. Neurofibromas can oftentimes be asymptomatic, but sometimes require surgery with a combination of chemotherapy. You can have malignant nerve sheath tumors, which have very poor outcomes. Trying to get extensive margins is often difficult, but surgery is the only major option. Lipomas can also present both ventral and dorsal, can incorporate surgical options. A lot of times these are asymptomatic. Paragangliomas are very rare. They can present more in like the lumbar sacral region. They're benign, but can be associated with severe neurologic deficits by time of presentation. Epidermoid cyst and dermoid cyst can occur within the spinal column. And then going into more of the intramedullary, we have astrocytomas and ependymomas being the most common. Ependymomas can vary on location and can be myxopapillary if more near like the conus. And then going on to other intramedullary tumors, we have ganglioma, which is a very rare tumor, has overall good prognosis, and hemangioblastoma is also very rare. Typically resection is indicated, and these patients tend to do pretty well afterwards. Here's the characteristics of overall performance status: poor, moderate, good. Number of extraspinal metastasis for bone mets. Number of metastasis within the bone. Metastasis to internal organs. Primary site of the cancer plays a role with increasing score as you get for rectum and thyroid cancer. And then overall complete, incomplete, or none in terms of palsies that are developed. So these give an overall score, and these can determine the overall prognosis. So the 0 to 8 prognosis less than 6, 9 to 11 can have 6 to 12, and then even further if it's above 11. The Harrington classification for spinal mets goes by class 1 through 5, and it is dependent on if neurologic involvement is severe, and also the potential for vertebral instability being the worst, so it's progressive as well. And then there's the SENS score, which incorporates location, looks at pain-free lesions, quality of the bone lesions, lytic versus blastic, and then also if there's kyphosis or scoliosis. This can result in overall like a degree of collapsement, and whether there's unilateral, bilateral, and extending throughout the columns as well. Overall, this can indicate when surgery would be an indication. Going into the molecular process, there is emerging immunosuppressive agents, specifically for those gliomas. So the astrocytomas primarily have been looked at more in the cranial literature, but there's ongoing work for spinal metastasis or spinal lesions as well, and drop mets for more of these gliomas. Still pretty much in its infancy, but having more and more data coming out. There's CAR T cell therapy, which has shown some early promise in terms of targeting more HER2 receptors, EGFR receptors. This can also play a role for intramedullary tumors within the spinal cord as well. And then vaccine therapies. This is using a natural, like viral mediated vector to recognize and respond appropriately. This can have emerging roles for a lot of the intramedullary tumors. So spinal tumors are rare compared to CNS lesions. They represent complex pathology and they're often difficult to treat. A lot of times surgery is the main option, followed by either radiation or chemotherapy, depending on tumor type. Biology of the tumors is ongoing in terms of the pathophysiology workup. A lot of emerging therapies are coming out in the cranial literature, and it will be necessary to continue research to apply this for spinal cord tumors as well.
Well, that's the video presentation by Dr. Brandon Lukeworth. For any queries regarding the presentation, kindly reach our program manager so that she'll guide you accordingly. By this, we come to the end of day one webinar. I would like to thank all the attendees and participants of today's webinar. Hope you enjoyed the webinar and gain knowledge from the seniors, and request you to attend tomorrow's sessions too. If you have any queries, you can mail us. Thank you. This is Shirley Rose signing off.